Put into focus. Okay, we're finishing off on the prostaglandins. A second for that to warm up. What's that?
Oh, you're waiting for that to warm up. I tried to tell her that Accutane was just an antibiotic the other day, and it was almost the same as taking vitamin A tablets.
Accutane?
Yes. That's what I thought. I'm like, I can, cause birth defects, and you can't go out in the sunlight.
Well, Accutane is nasty stuff. I mean, it works. It works. But it's...
She literally told me that they give it to their own kids, that it's just like, well, first, before telling her what it was like, I want to put you on antibiotics. And then she was like, oh, it's almost like taking vitamin A tablets. And I was like, well, then why can't you just take vitamin A tablets?
Like they're so big like vitamin A?
Yeah.
This is an MD?
Yes. That's scary.
I don't want to know.
I know. That's what I thought.
Accutane, I'm not quite sure how it works, but it is a nasty, it's a nasty, I mean, it works very well for acne. I know doctors that the anyway, I know that some doctors, family practice type, when they give Accutane, because the risks of birth defects are so high. I mean, you have to, by law, have a pregnancy test before you give it.
Every month.
But it can also cause problems, like adverse reactions with birth control pills, right? Because I knew somebody who was on birth control pills.
I'm sure it could.
What? Two birth controls when you're on Accutane.
Or the doctor will give you a shot of Depo-Provera when he gives you the Accutane.
And that is also excessively horrible for your body too, though, isn't it?
I don't know. They've never given me a shot of Depo-Provera. Yeah. My bulge was taken care of when they did the gastric thing. So. But I mean, to say it's like just taking an antibiotic or a vitamin is horribly misleading.
Okay, that's what I thought too. I was really, really vehement.
No, and there's more stuff coming out as to just how dangerous that drug
When you
is.
take it, you have to go maybe do exam quizzes on the website. If you don't have the exam, they won't let you.
Yeah, no, it's one of those things. It can do a very good job, but it's a very dangerous drug.
But there's always that should be a very last resort, right? It shouldn't be like, oh well, we tried one thing and it didn't work for a month, so we're going to put you on for acne. Yeah.
I would say it. Again, I'm not a doctor. I don't. I deal with the other end of it.
Right.
I do the birth control. And
tests that show all of those weird things that come up.
Yeah. Well, I don't see that much because they're very careful. At least the ones that I've had have been very careful. All I did was, my boss's daughter was on it, and when we did the pregnancy test, it had the negative. And I took a pen and filled it in and threw it on his desk.
Oh, you're honored.
And I still have my
Did you keep a straight face?
I did. Oh, yeah. I did. He knew I was messing with him.
Was it obviously a mark?
No. It looked like a regular positive.
A legit face.
Yeah. Okay. Just one more thing before we get off the prostaglandins. The NSAIDs. A few years ago, they came out with Vioxx and Celebrex, and this drives me nuts because you see it on TV, and ask your doctor if Vioxx is good for you, or Celebrex. I wish I would very much like them to cancel those types of ads on TV.
It's not good?
It tends to lead to strokes and cardiac arrest.
But they say it's better.
It's been pulled from the market. They've been pulled from the market. The good part about it is it was because it's a COX-2 inhibitor, it would act on some of the prostaglandins, but not on the stomach and not cause ulcers. But then they found that it did cause stroke and heart attack. So they pulled it. Avoid gastric, increase the chance of stroke and heart attack, so they have been pulled from the market. Now, the one thing that they didn't talk about here is the COX-3 inhibitor, which is actually extremely safe and used all the time. Tylenol. Yeah, acetaminophen. Yeah. Tylenol came out, well, I was in graduate school, so that would have been about 79 at that point. Tylenol was just coming out. It's a COX-3 inhibitor. Well, they knew it worked. They didn't exactly know how it worked or why it worked. The only thing with Tylenol is it's not anti-inflammatory. It's anti-pain and anti-pyretic. It'll knock the fever down. It'll knock the pain down. It will not knock the inflammation down. So if you've got, if you've got a sore ankle or something from exercising, you might want to take the ibuprofen to knock the inflammation down, but then also combine it with Tylenol. So take 500 Tylenol and 600 or 400 or 600 ibuprofen. So it not only cuts down the inflammation, it also does a better job of cutting down the pain.
But why do they say it's used to treat asthma? It has nothing to do with...
Yeah, Tylenol?
It's said here in the book. I'm sorry, it's in another... I'm sorry.
What does it say to use?
The antinucleotrients have recently become available.
Well, yeah, because some of your prostaglandins are vasodilator. And that's what would be in your inhalers to, like your... Singular.
Yes.
That's a vaso or bronchodilator, excuse me. And that's what they would use to treat asthma.
What is ibuprofen?
What's that?
What is ibuprofen exactly? What does that do? Why is it so bad for you, you said before?
It's bad because it causes everything that the NSAIDs cause. It causes the kidney problems. It causes the gastric bleeding, just kind of like aspirin. So I think they're probably getting pretty close to banning it in Europe, the ibuprofen.
Do you think they will here really?
No, the military likes to give it out too much. No. They give it to you. The joke was vitamin M. You go in for anything, and they're like, "Oh, here's a bottle of Motrin."
Gee, I thought it was Tylenol. Light fighter's candy.
Well, my kid's doctor said now they found that Tylenol also causes some sort of...
Well, anything in it.
Liver damage for kids, right?
Yes.
Oh, well, Tylenol or acetaminophen has very much, and this is one of the things that I did in the paper that I did back all of those years ago. Tylenol is very much implicated in liver problems. People would use Tylenol to commit suicide, so they would take it. They would take a toxic dose, and then they would wake up in a few hours and say, "Oh my God, what did I do?" And then come into the ER and say, "I took 10,000 milligrams of Tylenol," and their liver's shot. They can't do anything about it. Sometimes they can put them on a mucomist, which you use actually for pneumonia in kids. Huge amounts of that. Sometimes it'll help, but a lot of times, you'll put $100,000 into therapy and they'll still die.
So what do you use if you have?
I use Tylenol. You keep your Tylenol down under 4,000 milligrams a day. That's 2 extra strength Tylenol 4 times a day. You can do that, and you're okay, unless you have a liver problem to start with, and then you just cut it down from there.
And why not Advil? You just said Advil is...
That's ibuprofen.
That's ibuprofen. So you still have the gastric problems. Me, after my stomach surgery, they say, yes, I can take Advil, or ibuprofen, or what's the 12-hour one? Aleve. But they highly discourage it. because it does cause stomach bleeding and stomach problems. But again, on a short-term basis, to relieve the inflammation of a sprained ankle or something like that, you just can't take it on a regular basis.
So is aspirin really the best, probably, even though it messes your stomach, for pain?
Well, for pain, Tylenol would be better than aspirin.
Inflammation is ibuprofen.
Inflammation, aspirin would work better on inflammation. Obviously, Tylenol would not work on a bee sting. On a bee sting, you take as much aspirin as you can tolerate because it cuts down the prostaglandin response. It makes the recovery tremendously easier.
You can also put it into a paste and apply it topically.
Works, it kind of works that way, but stuff like the Aspergum doesn't work at all. It gets down into your system, then it comes back and works. But yes, you can put an aspirin paste on a bee sting. The time that it was done to me, I had gotten about 30 hornet stings. I'd stepped on a hornet's nest. Got me
More dangerous stuff for kids to take the Tylenol?
No, kids. It's actually more prescribed for kids for fever reduction.
It's a weird.
You just don't take it in the dose. You take it in a smaller dose.
Tylenol won't. I don't know why, but Tylenol won't break the fever at all. And my daughter runs high, like 105 fevers usually, and it doesn't do a thing. It's like giving her placebo, except for ibuprofen will bring it down.
I would check the bottle labels. Ibuprofen isn't supposed to, and Tylenol is. No, I'm giving you bad. I don't know. I just simply.
Well, I can talk to the pharmacist.
Yeah, but the Tylenol is supposed to be a much better anti-fever medicine.
Maybe it's just her.
Could very well be. Okay, and that I believe is it for chapter 11. So, taking 2 seconds to talk about the test.
Instead of taking the last time, that'd be great.
No. Just a word of advice as you start looking at this stuff. We talked about, the adrenal medulla. We talked about the epinephrine. We talked about parasympathetic systems. We talked about the sympathetic systems. It's all in your notes. What I'm saying is when you review for the test, one thing, and I do have most of the questions written, there are going to be some specific things like on autocrine or parasympathetic or sympathetic. You're going to need to know the parts of the brain, have a general idea of where your cranial nerves and all that are coming from and going to. Most of them come out of the medulla oblongata. Your vagus nerve is what? 10th cranial. I'm asking that because that's the only one I remember without looking at the book. But a lot of what I'm going to be looking at, probably at least 50% of the test, is things that are going to be in common with, you've got 5 chapters that are going to be in common with 3 or 4 of them anyway. We touched on the adrenal cortex. We touched on it with the tissue. We touched on it with the parasympathetic. We touched on it with the epinephrine. We touched on it, with the hormones. Stuff like that. There's going to be, some questions like that on there. So just keep that in mind, and we will do some reviews. I've got about 100 questions for the test, and I've pared that down a little bit. So.
What do you mean we'll do some reviews?
Probably if we have time after the lab on Thursday, this coming Thursday, I'll spend a few minutes kind of going over some of them.
Everybody finishes at a different time, I think.
Yes,
Look, I'm flying out at 6 o'clock the next morning.
You have to do it before.
No, because you got to finish the lab.
Yeah, we'll go off. It'll be all right.
Yeah, you leave early.
What labs are going to be on the test?
We did 1 through 4.
Correct.
So we're going to have hearing, vision, the sensory, the vision, and the cellular metabolism. Because I think we tested up to cellular metabolism, didn't we? You're going to look at probably the next 4 labs after the ones, we did 1 through 4. 5, I think, was cellular metabolism.
Yeah.
So you got 5, we didn't do digestion. 6, we did
5, 10,
9, 10, and 11.
We did 11 just last week.
Yeah, well, 5 through 11 then, because you'll get 11 back. You should have gotten 11.
So 4 labs.
4
11's
labs.
due tomorrow.
11's due tomorrow.
So-
We'll go over the answers on Thursday. Okay? Okay. So I'm gonna run you a couple of videos that just very quickly go through the contraction, the anatomy of skeletal muscle of muscles, and how they work. We'll start with this one.
The skeletal muscle of animals, including humans, are striated. They exist in bundles of thousands of muscle fibers. A single muscle fiber is actually a muscle cell. As you can see, muscle cells are very elongated and contain several nuclei. Packed inside the muscle cells are contractile myofibrils. Myofibrils show the striated pattern as repeating subunits called sarcomeres. When a motor neuron stimulates muscle fiber, overlapping filaments in the sarcomeres slide along one another, and the sarcomeres shorten. The repeated pattern of the sarcomere tells from the arrangement of the two major proteins within the myofibrils. Myosin and actin. The myosin test is the actual mechanism for muscle contraction. Now let's examine the process in detail. First of all, when a motor neuron stimulates a muscle fiber, calcium ions are released from the sarcoplasmic reticulum. The ions force the tropomyosin to displace and expose myosin binding sites in action. An ATP molecule is unhydrolyzed, and the myosin head forms a cross bridge. When the ADP is replaced by a new molecule of ATP, the myosin head detaches from actin and goes back to its original state. And that's pretty much the whole process. And one more thing. Each myosin head attaches to and releases from actin independently, not in unison. Otherwise, the myosin head would all release at once, and the whole muscle would go to a relaxed state. And that wouldn't be good.
Give me a second here. That's
Tells you everywhere. Please
what was that?
The way that I had the muscle reaction taught me so I would remember it. Calcium is a fairy godmother. And it comes down, and the actin is an evil stepmother who won't let the triple myosin and myosin hang out with each other. And so it reacts with the actin so that it gets out of the way, so that, and then that also pulls the troponin out of the way because the actin and troponin, I don't know, are...
Pulls the tropomyosin out.
Really, is that... Because tropomyosin binds with the myosin, I thought. The troponin gets moved out of the way.
Troponin gets moved out of the tropomyosin binds with
Yes,
the
so the
myosin, with the
actin-
myosin head.
So the calcium is a fairy godmother that gets rid of the evil stepmother out of the
The
way.
calcium
Binds
elevates
with
the
the
amount
myosin.
So I didn't mean to do that. Hang on a second.
the lecture.
Okay, time to go home.
Yeah.
Turn around.
Why are you such false advertising?
I know, that's so mean. You're like a mean
Yeah.
older brother.
I'm like that.
You're an older brother, aren't you?
I'm like the troponin there. Evil troponin.
Was the father of the story?
Because we don't want to be that far behind.
That Khan Academy has some videos.
Your goals for learning are to compare and contrast smooth muscle cells, cardiac muscle cells, and skeletal muscle cells. To review the anatomy of the skeletal muscle. To examine the connective tissues associated with skeletal muscle. To review the intracellular organization
I
of the
don't know
skeletal
where
muscle
they
cell.
had a diesel truck in the background when they recorded this.
The definition of connective tissue. The
Because you
structure
can't
of
put
a
on my
typical cell.
laptop the same way.
To see definitions of terms, click the bold red words. The contractile cells of the body can be classified into three major groups: skeletal, cardiac, and smooth muscle cells. These three types of cells can be distinguished on the basis of their shape, number of nuclei, position of nuclei, presence of striations, and whether they are under voluntary or involuntary control. Skeletal muscle has elongated cells with multiple peripheral nuclei. Notice the striations, the white and dark bands, in the photomicrograph. Later we will explore the details of this banding pattern. Skeletal muscle cells are under voluntary control. Cardiac muscle has branching cells with a single central nucleus. Cardiac muscle cells are also striated. Unlike skeletal muscle, cardiac muscle cells are under involuntary control. Smooth muscle cells possess a single nucleus within a spindle-shaped cell. These cells do not have visible striations. Like cardiac muscle, smooth muscle is under involuntary control. The focus of this module will be on the physiology of skeletal muscle. Here we see the brachialis, a skeletal muscle attached to the humerus and ulna by its tendons. For a cutaway view, click the muscle.
It does it for you.
Let's look at the internal structure of a skeletal muscle. A typical skeletal muscle is composed of an orderly arrangement of connective tissue and contractile cells. The entire muscle is surrounded by an external connective tissue wrapping called the epimysium. The skeletal muscle itself is made up of fascicles, which are bundles of individual muscle cells. Each fascicle is surrounded by a connective tissue layer called the perimysium. Click the fascicle to enlarge it. This view provides a closer look at the internal structure of a fascicle. Recall that fascicles are bundles of muscle cells. Within the fascicle, the third connective tissue layer, the endomysium, separates and electrically insulates the muscle cells from each other. All three connective tissue layers, the endomysium, the perimysium, and epimysium, bind the muscle cells together, providing strength and support to the entire muscle. The connective tissue layers merge at the ends of the muscle and are continuous with the tendons. To continue moving through the levels of skeletal muscle, click on the muscle cell to enlarge it and view. Because skeletal muscle cells are elongated, they are often referred to as muscle fibers. Let's look at the internal structure of these unique cells. The nucleus is the cellular structure that contains the genetic material.
Notice where it is.
The sarcolemma is a specific name for the plasma membrane of the muscle cell.
Space between
The sarcoplasmic reticulum, or SR, is the name for the endoplasmic reticulum of the muscle cell. Its interconnecting tubules surround each myofibril like the sleeve of a loose V-neck sweater. Terminal cisternae are sac-like regions of the sarcoplasmic reticulum that serve as specialized reservoirs of calcium ions. T-tubules are inactivations of the sarcolemma projecting deep into the muscle cell's interior. Note the visible T-tubule openings on the surface of the sarcolemma. A triad is a three-unit group consisting of one T-tubule lying between two adjacent terminal cisternae. The cytosol is the intracellular fluid in which the organelles are suspended. Mitochondria are cytoplasmic organelles that are sites of ATP synthesis. They are referred to as the powerhouses of the cell. A myofibril is a cylindrical bundle of contractile filaments within the skeletal muscle cell. To review any of these terms, click on the bold red words. Click the myofibril to enlarge it and explore its composition. The myofibrils that are packed into a muscle cell are composed of individual contractile proteins called myofilaments. There are two types of myofilaments. The thin filament is composed mainly of the protein actin. The thick filament is made up chiefly of the protein myosin. Details of thick and thin filament structure are covered in another topic, the sliding filament theory. To examine the arrangement of myofilaments, click the myofibril. The arrangement of thick and thin myofilaments forms light and dark alternating bands along the myofibril. This creates the cell's striated appearance, as shown here in this photomicrograph and diagram. Features of the light and dark bands are identified by letters. The dark bands are called A bands. The A band corresponds to the length of the thick filaments. The light bands, called I bands, alternate with the dark bands. The I band contains only thin filaments and corresponds to the distance between adjacent thick filaments. During muscle contraction, the I band gets narrower. Each I band is bisected by a zigzag line called the Z line. The Z line is actually a protein disc which anchors the thin filaments and connects adjacent myofibrils. In the middle of each A band, a lighter stripe appears called the H zone, corresponding to the region between the thin filaments. The width of the H zone varies depending on the degree of muscle contraction. It is widest when the muscle is relaxed and stretched. The M line, located in the center of the H zone, consists of protein fibers that connect neighboring thick filaments. The region of the myofibril between two successive Z lines is called a sarcomere. It is the contractile unit of a muscle cell. You will see how a muscle cell contracts in a later topic, the sliding filament theory. Now that we have taken a skeletal muscle apart, let's put it back together, starting with its smallest building blocks, the myofilaments. There are two types of myofilaments, thick and thin. Many of these myofilaments bundled together make up a single myofibril. Numerous myofibrils are contained in a muscle cell or muscle fiber. In turn, many muscle cells are packed into a fascicle. And finally, many fascicles make up a whole skeletal muscle. This bundle within a bundle organization is well suited to the function of skeletal muscle, as we'll see throughout the module. Click a link button to review a structure in more detail. If you use a link button, you can return to the page. The arrangement of thick and thin myofilaments forms light. Now that we get in summary, a whole muscle can be viewed symbolically as a pyramid of subunits. A whole muscle is composed of many smaller units called fascicles. Each fascicle, in turn, contains numerous muscle cells, also called muscle fibers. And as we have seen, each cell is composed of many additional subunits called myofibrils. The smallest and most numerous of all subunits are the myofilaments, which are found within the myofibrils.
They are in alphabetical order, as a trick.
Here's a summary of what we've covered. The three types of muscle cells in the body are skeletal, cardiac, and smooth. Skeletal muscle has 3 layers of connective tissue: epimysium, perimysium, and endomysium. The striations of skeletal muscle cells are due to the organized arrangement of contractile proteins called thick and thin filaments. A whole muscle demonstrates a bundle within a bundle organization, from myofilaments to myofibrils to muscle cells to fascicles to the whole muscle. To access cross-references for this topic in your Benjamin Cummings textbook, click here.
What was the name for this one?
I went in and I put in muscle contraction and structure. It's Anatomy and Physiology Review of Skeletal Muscle Tissue. That's the name for it. This is a quick one on the sarcoplasmic reticulum. The sarcoplasmic reticulum is again the plasma membrane, essentially. The endoplasmic reticulum, sarcoplasmic reticulum is the same type of structure as the endoplasmic reticulum in a normal cell. In the muscle, the sarcoplasmic reticulum stores the calcium. So when the nerve activates it, that's where the calcium comes out and allows the contraction to take place. If I get a chance to. This one, so what you're seeing here, and I don't know if there's any sound of this. And obviously, since it's cardiac, it's probably not. But this is the calcium.
On mute right now.
It's a little hand
So here's the heads pushing the troponin out of the way. They're binding, moving the muscle fiber. Kind of different.
To activate skeletal muscle, the central nervous system initiates an action potential that travels down the spinal cord to the motor neurons. As the nerve fiber branches, the action potential travels down each branch. Each nerve fiber branches many times and stimulates several skeletal muscle fibers. To activate skeletal muscle, the central nervous system initiates an action potential that travels down the spinal cord to the motor neurons. As the nerve fiber branches, the action potential travels down each branch. Each nerve fiber branches many times and stimulates several skeletal muscle fibers. The union of the axon and muscle fiber is called the neuromuscular junction. Zooming into the microscopic level, each branch of the neuron has a terminal that invaginates the muscle fiber while remaining outside the muscle fiber plasma membrane. The action potential arrives at the axon terminal. In the terminal, the action potential causes the release of acetylcholine from the synaptic vesicles into space between the axon terminal and the muscle fiber, called the synaptic cleft. In the synaptic cleft, the acetylcholine binds with a receptor site on the fiber membrane, which opens a chemically gated ion channel. Sodium then rushes through the ion channel into the muscle fiber, causing an action potential to form on the fiber membrane. The action potential spreads along the muscle fiber. As more nerve branches activate additional fibers, the action potential spreads over the entire muscle. Upon activation, the muscle contracts.
That wasn't the one I was looking for. So see, now we're going to look at the sliding filament. How much fun did you think you were going to have?
Oh, yeah, it's done.
No, we are not. It's going to be done.
No.
This is just the. This is what you're going to be
We
testing.
move our bodies by contracting skeletal muscle. While the results of this action are plain to see, the mechanism behind the action is not.
Can you turn it up a little bit?
Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten.
So
It is
come here
important to
between
note
the
that the
two
individual
lines.
myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached to the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with their troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
The calcium, hang on one second. The calcium, by active transport, is pumped. There are calcium pumps that pump it back into the endoplasmic or sarcoplasmic reticulum. And it's just, it's recycled at that point. Okay. Mechanisms: muscle control, contraction, neural control. We start out with a lot of it. What you're going to see is what you saw in the videos. And those videos are that just Google putting in muscle contraction, Google putting in muscle contraction, structure, function. Neuromuscular junctions. When an axon goes into a muscle, it goes into a neuromuscular that's called a neuromuscular junction. It goes into a neuromuscular motor plate. Now, a motor unit, because I believe that's on your homework, a motor unit is one nerve and all the muscles that it innervates. That's a motor unit. Muscle contracts, and muscles do not extend. A muscle cannot push. The only thing a muscle can do is contract. So you have antagonistic muscles. You have extensors and you have flexors. Different muscles moving in different directions. Muscle contracts, it shortens, it places tensions on the tendons that are connected to the bone. You saw that. Moves the bone at a joint. The bone that moves is attached at where the muscle or the tendon goes in is a muscle insertion site. Attached to the bone, it does not. The muscle is attached to a bone that does not move at the muscle origin. So you've got one bone that's going to move and one bone that's not going to move. That's why you get the flexation or extension. Flexor muscles decrease the angle between two bones at a joint. It's like this. The angle is 180 degrees right here. 90 degrees. Whatever. Extensor muscles increase the angle. Muscle responsible for movement in a given direction is the agonist and the antagonist. Flexor and extensors are antagonists. So you have the agonist, which moves it in the direction, the antagonist would move it back. Extensor increases the angle at a joint. Flexor decreases the angle at a joint. Abductor moves the limb away from the midline of the body. Adductor moves it back. Levator moves it upwards. Depressor moves it down. Rotator rotates the bone on its axis. A sphincter constricts an opening. Skeletal muscles are surrounded by epimysium, which we saw. Connective tissues called the perimysium divide the muscles into fascicles. Fascicle is divided into muscle fibers and surrounded with the endomysium. And you saw pictures of that. Here is the structure. Periosteum covering the bone, the tendon going into the bone, fascia, skeletal muscle. Epimysium, perimysium, endomysium. Fasciculus, muscle fibers. Here you have the striations, which are the dark and the light bands. Obviously, the striation does not go through the entire muscle. It's just how the fibers are stacked up. Nuclei are on the outside of the muscle fibers. They are in the endomysium. I'm sorry, not in the endomysium. They're in the perimysium, in between. There's no room inside for the nucleus. And then you have your muscle filaments and myofibrils. Muscle fibers, plasma membrane is called the sarcolemma. They're multinucleated because they're formed from many stem cells. They're striated. The I bands are the light bands, the A bands are the dark bands. There's your nuclei. There's a muscle fiber. Neuromuscular junctions and motor end plates. The neuromuscular junction is where the neuron, the motor neuron, comes in. Is it afferent or efferent? Is the neuron afferent or efferent for a motor neuron? Afferent. Going away. Efferent. Going away. Afferent's coming in. Efferent. It's going from the spinal column out. Motor end plate. It's the area of the muscle fiber or the sarcolemma where the motor neuron stimulates it. Now, did she talk about the T tubules?
No.
Okay. On your we'll see it here. Oh, the girl that was in the video. The girl voice. Somatic neuron axon. Mitochondria, I think they pretty much showed you this one. Nerve fiber branches coming down. There's your motor end plates here. Your myofibrils. Here's your motor end plate right here. Your synaptic cleft. Neuromuscular cleft, same deal. Synaptic vesicles. What do they contain? Acetylcholine. A single motor neuron and all the muscle fibers it innervates as a motor unit. All the muscle fibers in a motor unit contract at once. Now, depending on the degree of control of the muscle, you will have more neurons, or excuse me, less muscle fibers per motor unit than a very big muscle that does not need a lot of control. For instance, you're gonna operate on an eye. You don't hold the scalpel between your toes and try to operate. In your hands, you'll have much fewer muscles per motor unit because you'll have much more delicate control. Your eye, you need to be able to look at, to control the movement of the eyeball. You have somewhere between 5 and 20 muscles per nerve. On the leg or the back or the thigh, you might have 2,000 muscle units per nerve. So that nerve is going to come down, it's going to branch, it's going to innervate, that one nerve is going to innervate 1,000 to 2,000 muscle fibers. So you don't have nearly the amount of control. Motor unit coming out of the dorsal root. The somatic motor neuron, somatic means that it's voluntary. And you can see that the motor unit is innervating these muscle fibers. Neuromuscular junctions, skeletal muscles, this is your motor unit. The nerve comes in and it will innervate however many muscle fibers, somewhere between 5 and 2,000, I think they say. Contraction strength comes from the motor unit recruitment. What happens is as you require more strength, more motor units fire. Once it fires, it's an all or nothing. So you recruit more motor units, more muscles to fire to do what you need to do. Finer muscle controls requires smaller units, fewer muscle fibers. This says 20, I've seen it as low as 5. Larger, stronger muscles have motor units with thousands of muscle fibers. So when that fires, all those muscle fibers are firing. Control and strength are trade-off. Mechanisms. Muscle fibers, densely packed subunits called myofibrils, run the length of the fiber. You have the thick and thin fibers. Again, you've seen it. Sarcolemma, sarcoplasm. Sarcoplasm is like your plasma membrane. You have your myofilaments, you have your myofibrils. And then inside you have the myofilaments. Striations are the overlap between the thick and the thin bands. Striations produced by the thick and thin filaments. I bands are the thin. A bands contain all of the thick with some thin overlap. H bands are in the center. They have no thin filament overlap. Z lines are made of the discs that go right down the middle that kind of hold the filaments together. M lines are in the center of each A band and help hold down thick filaments. Z lines are for the thin filaments. Sarcomere. The muscle of the sarcomere is from one Z line to the next. Titin is a protein that anchors the thick filaments and allows elastic recoil. Now, when you look at muscles, say in the diaphragm, you have the muscles that expand it. But it's when you relax, it's this elasticity that allows it to come back and allows you to breathe. You lose that elasticity, you have much more trouble breathing. So here, You've got your Z disc. You've got your cross bridges with the myosin. You have your A band, your H zone, your clear zone, your thick filament, which is what? Myosin. What's your thin filament? Actin. Now, you have your myosin heads here. In the book, it describes it as like cocking a pistol or pulling back a bowstring. You have the energy stored in that. So when that ADP breaks off, you have your power stroke. So it's either the hammer coming down or the release of the bowstring. You know, the energy is stored in the myosin head in that phosphate bond. So here is the direction of movement in a contraction. Here's your myofibril. Look at all the fibers. Here's the sarcomere between the two Z lines. Sarcomere shorten. The bands do not shorten. The filaments overlap. The A bands don't shorten. The I bands, the light bands shorten because the thin filaments are overlapping. They slide over the thick filaments. They're actually pulled along by the myosin heads, like you saw in the video.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
Okay, thick is composed of myosin. It has ATP, two globular heads, ATP and actin binding sites. Thin is actin. Proteins called tropomyosin and troponin prevent myosin binding. In the lab, the new test, not the new test, it's been out for a few years, is when we look at cardiac damage, we're now looking at troponin. Troponin I or troponin T released from the muscle cells of the heart. It's much more sensitive than the CK, much more specific. Okay, monofilament structure. Here is your myosin head, your attachment for ATP that is converted to ADP and a phosphate. That's the tail. Here's your actin. You have your troponin, you have your tropomyosin. These block the action until what gets there? Calcium. Calcium's released from sarcoplasmic reticulum, where it's stored. When it's done, so another thing, just to be aware, you're using an ATP here, so you're using energy as part of each power stroke. Also, to get the calcium back into the sarcoplasmic reticulum, it's an energy requiring pump. So you're using more ATP there. So when you're doing a lot of muscle work, you're using a lot of ATP. Sliding produced by cross bridges that form between the myosin and the actin. Myosin head serves as a myosin ATPase, splitting ATP to ADP. So the myosin head is acting like an enzyme. This is the myosin head here. It's got ATP. The ATP allows it to cock back. When the ATP is broken off to ADP, you get your power stroke. And it just grabs it little bit by little and pulls it together. Myosin serves as ATPase, splitting the enzyme into ADP. It binds to the actin, when the muscle is stimulated. Upon releasing of the phosphorus, it cocks and produces a power stroke that pulls the thin filament toward the center. So, I don't know if you still got them. Russell had PVC, one thick, one thin. You can slide them together just to stimulate a muscle action. But it's the same thing. You've got the filaments around each other. You've got the myosin heads. They're essentially just reaching out, grabbing, attaching, and pulling it through. Cocking back, attaching, pulling it through. Probably to move they say it can move the actin about 1% per stroke, and it'll contract to about 60% of its total length, the sarcomere. Now, these do not work in concert. They're not timed, or else you would have, it wouldn't be, you couldn't have a constant movement because all of them would fire, and they'd stop and regroup, and then they'd fire again. So they're working separately so that you can get a smooth muscle contraction. Cross-bridging. After the power stroke, the ADP is released and a new ATP binds. This makes the myosin release from the actin. You get to cock back. The ATP is then split. The myosin rebinds further back. You have the ADP and the phosphorus. You have your power stroke again. Resting fiber. Cross bridge is not attached to the actin. You have ADP, but it's not attached. Cross bridge binds to the actin. There goes your phosphorus right there. Phosphorus is released from the myosin, causing conformational changes. The power stroke causes the filaments to slide. New ATP comes in. The ATP is hydrolyzed. The phosphate binds to the myosin, causing it to cock again and getting ready to go. And it just goes round and round and round. As long as there is calcium. If there isn't calcium, the whole thing stops. Tropomyosin blocks the cross bridges. Troponin inhibits the binding of I, inhibits the binding of myosin. T, tropomyosin. C, tropin C binds to the calcium. There's your complex. When muscle cells are stimulated, calcium is released inside the muscle fiber. The calcium is stored in the... You guys are going to get that. I'll put it on the test just to... Well, it'll be the next test. You'll forget it.
You will too.
No, I won't. No, of course not. Calcium, released from the muscle fiber, attaches the troponin C, causing a conformational change in the troponin and tropomyosin. The myosin is allowed to access and form cross bridges. The calcium, like you said, Sarah, gets it out of the way, allows the myosin to attach, and then you can have your power stroke. There's your troponin, there's your ATP, your calcium comes in, and and this is the thing that we've been talking about ever since we started about cellular structure and function. When something attaches, it causes a conformational change. For an enzyme, your coenzyme attaches, it causes a conformational change. Very much.
Thank you.
He thinks he's getting this one back. I don't think so. Okay. All the things that we've talked about. Your neurotransmitter comes across the synapse. It binds with a receptor. It causes a conformational change. It opens up the sodium gates. Kind of get a theme here? This is how a lot of things work.
You probably took the batteries out.
No, because
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
sarcoplasmic reticulum. It's a modified endoplasmic reticulum that stores calcium. It's stored as it showed you in the terminal cisternae, and you have it one on each side of your Z line. This is going to be confusing. Okay. Okay, sarcolemma, terminal cisternae. Your transverse tubule. The transverse tubule is a tubule that goes through the sarcolemma, goes into the middle, in and around the myofibrils. It allows the calcium to get in where it belongs. It's a pathway for the calcium to get in there and cause, get the troponin and that out of the way so that you can have your power stroke. Okay. When a muscle fiber is stimulated, calcium diffuses as out of the calcium release channels in the sarcoplasmic reticulum. At the end of the contraction, calcium is actively pumped back in. So you're using more energy to get the calcium back. You're using ATP for the power stroke. You're using more ATP to get the calcium back in. Transverse tubules. Narrow members, tunnels formed from the sarcolemma. Open to the extracellular environment. They conduct action potentials. They let the calcium in. They're closely situated next to the terminal cisternae. When the calcium comes in, it travels through the transverse tubules to get into the muscle. Okay, stimulating a motor fiber. You have acetylcholine from the motor neuron. The end plate potentials are produced. The acetylcholine comes back across. It binds to the receptors. It opens up. You get the what in and the what out. Sodium in, potassium out. Its action potentials are generated. The action potential is a voltage gated. So it's not a chemical gated. So what happens is your membrane potential is dropping. It goes from a minus 70 down to a minus 50, minus 40. This causes the sarcoplasmic reticulum to open up and release calcium. Calcium goes in, it knocks the troponin out of the way, binds with the troponin, moves it out of the way. The myosin heads can attach, and you have your power stroke, you have your muscle contraction. Okay, somatic motor neuron, acetylcholine, ACH is released, binds to the nicotinic ACH receptors, open ligand, a chemical gated channel. So it's at the synaptic cleft. It's not an electrically gated channel, it's chemically gated. It's the binding of the acetylcholine to the nicotinic receptors. Sodium diffuses in, depolarizing an action potential. The action potential goes along the transverse tubules. It opens voltage gated. The voltage membrane is dropping. It opens the voltage gated gates and channels, and calcium comes out. Calcium gets into is released from the sarcoplasmic reticulum. It diffuses into the sarcoplasm. It binds to the troponin, stimulating contraction. Here's another diagram. Same thing you saw in the previous one and in the video.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles. An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
Muscle relaxation. Calcium's, the action potential is there. It's not releasing calcium. Calcium's being pumped back into the sarcoplasmic reticulum. It's not there to allow the cross bridges. I think that's enough. Not that you haven't had enough movies. I'm not sure that I have enough of these to go around. Here is for the lab on Thursday. This is we're going to be using the computers and the simulators. So if you can share these a little bit. ahead, take that. Okay.
Oops, I'm sorry. Thank you.
Oscar? So if you can,
Do an airport thing on Friday. I'll take the rest.
there you go. Now, do you kind of see how we're starting to go back over stuff that you've already gotten in the basics?
I'll make copies for them.
I've got 4 or 5 left, so. You need one, right? Andrew, you got one?
I already got one, thank you.
Okay. You got one, Oscar, right?
Thank you. Don't go to the Gilroy kiosk. It's not good.
What?
Don't go to the Gilroy kiosk. Alicia said you're going to the kiosk.
Oh,
The
I
Gilroy
go to the one
one's
out on Monterey.
Oh,
Where
yeah,
the Shell
that
Station
one's fine. The one by Napo is every time.
That's
they-
downtown. I never go down here.
Yeah. it's
Never go
fine.
down there.
The one in Morgan Hill's good too, right off the freeway.
Yeah. I used to know the guy that owned that. But,
Doesn't he still own it?
oh, this was 20 years ago.
Yeah. They still own it. It was a couple. Older couple.
Well, they are now, I suppose.
Well, they were then.
I think he's one of my son's coaches. No, I'm not sure.
Their picture was in there for a long time, but.
No, it wasn't the same guy then.
Okay.
Same guy. But no, KFC is something that I will sneak when I'm 2 states away so I won't get caught. That's
No, you
not
just
how it works.
throw away the stuff. Smells on you. You throw away the stuff before you go home, that's all.
Yeah.
I do the same thing to my husband though. That's just because your wife cares about you.
Just once in a while. Have a good evening. We'll see you Thursday.
Okay, bye.
Okay, you guys got the paperwork? Okay,
Yes.
good.